Hi, welcome to lecture on environmental adaptation for animals. Now we will focus on the other side of the respiration, not to maximize the oxygen that we get in the body, but how to get rid of all the carbon dioxide. And we have to remember first that the carbon dioxide is much more, it's easy to dilute in the water versus the oxygen itself. And still, although we can have high concentration of carbon dioxide in the blood, we only carry 5% of the carbon dioxide as the carbon dioxide molecules. So if we have in any aquatic solution, add the carbon dioxide, it will form carbonic acid. It will react with the water. And this reaction is not very efficient, so only little bit carbonic acid is formed. And then this carbonic acid is easily dissociated to form the carbon dioxide back. Okay, so you don't have to worry on that. But this carbonic acid is reacting because it's an acid, it has 2 protons. If it leaves 1 proton, then it will form the bicarbonate and dissociate leave a proton that will affect on the pH. And this equation is, this reaction is very good. So a lot of bicarbonate is formed versus the synthesis of carbonic acid is quite slow. And therefore you can have high concentration of bicarbonate. And in some cases, this bicarbonate is even reacting or dissociated, so it has still 1 proton left. It can leave it and form the carbonate. So instead of having a single carbon-containing molecule that you are having in your circulation, you have another 3rd and 4th molecule. So you have 4 molecules that are reacting, and they can be modulated to form these others. And what about then in different partial pressures? So what about then when having low concentration, so low metabolic rate? So you have oxygenated blood, and then after it goes in all these cells, and they are giving the carbon dioxide to carry. So first of all, we have over here the partial pressure of carbon dioxide, and over here we have the concentration of different these molecules. And this is a logarithmic scale. So this is a millimole per liter, this is a micromole per liter, this is a nanomole per liter, this is picomole per liter. And let's look first on the carbon dioxide and the carbonic acid. This black line is much lower than the blue line. And this difference is about 1000 times, because it's a logarithmic scale. But then we have the bicarbonate. And this difference is something like 100 times or 1000 times higher. So that means that we have carbon dioxide and bicarbonate in the circulation. And then the 4th molecule, the concentration is anyway very low. Especially in the physiological partial pressure of carbon dioxide, we only want to worry on these 2 molecules. And if we are forming from the carbon dioxide a lot of bicarbonate, we will affect on the dissociation of this acid to leaving a proton that is affecting on pH. But first, let's look that, okay, the carbon dioxide concentration is increasing. It looks like it's increasing linearly, but because it's a logarithmic scale, so actually it's rising exponentially. And therefore, in very high partial pressures, all molecules are as a carbon dioxide. but because there's also the production of carbonic acid, bicarbonate, carbonate, etc. So there's dissociation in which you are leaving protons that are affecting on the pH will drop. And this is the physiological range. So you can see that high partial pressure of carbon dioxide, you will have lower pH if you are not doing anything on it. Okay, so what if we keep the pH constant? How to keep it? First of all, have a beaker. So here we have water, and in the atmosphere we have partial pressure of carbon dioxide is 0.04 kilopascals, and it will dissociate it. It will be dissociated in the water until the partial pressure in the water is the same, so they are in the equilibrium. And now this carbon dioxide will form partly the bicarbonate, and we can put this reaction a little bit easier because we don't want to worry on the acid that was there between, because it doesn't exist a lot. So now on the left hand side we only have carbon dioxide, water. On the right hand side we have bicarbonate and the protons that are affecting then of course on the pH. And we can always, from these equations, these reactions, we can calculate the equilibrium constant, in which the concentration of these products is divided with the concentrations of these molecules that were producing these. So the concentration of bicarbonate, then the concentration of protons, okay, that's kind of pH thing. Then the concentration of water and the concentration of carbon dioxide. Well, we don't have to worry so much on the differences in the water concentration, because there is plenty of water anyway. And also this dissociation constant is, we can keep that, okay, it's always the same. And therefore we can understand that, okay, then this amount of bicarbonate is depending on the carbon dioxide, it depends on the dissociation constant, it depends on the pH. And therefore, if we are having these protons, concentration goes down, then this will go up. And how we can put these proton concentration down? So it's used with the pH buffers. So they are binding the protons, and therefore the concentration of free protons are low. And our body is very good in catching these protons. So we are doing it in two ways. We are binding it in the N-terminus of proteins, and we are binding it in the histidines. N-terminus is in all proteins, so N and then we have amino acid, and then we have the C-terminus. And over there we can bind one extra proton. And in some cases we also have one amino acid called histidine, which can also bind a proton. Okay, and how we can do this in the blood? What is the most common protein there in the blood? It's hemoglobin. Okay, but still, depending on the partial pressure of carbon dioxide and depending on the concentration of bicarbonate, it affects on the pH. According to the Henderson-Hasselbalch equation, the high partial pressure is lowering the pH, but you can see that this difference is not so huge anymore because we have these buffers. And also, if we are having more carbon dioxide formed in the molecule of bicarbonate, then the pH is rising. If the partial pressure is all the time the same. Okay, so that's why we want to have as much carbon dioxide in the form of bicarbonate in the blood as possible. So what about then in the circulation? When we are having a higher partial pressure of carbon dioxide, we will have higher concentration of carbon dioxide. But this is in all forms. So this is actually the concentration of carbon dioxide itself, and this is much higher. It's mainly in the form of bicarbonate. But then a small part of it can be binded also in the amino terminus of the hemoglobin and other proteins forming carbamate as a carbamate group. So this N group is binding not only protons, but it's also binding carbon dioxide. But still, the bicarbonate is the main product. And therefore, the speed of this reaction to produce these bicarbonates is affecting on the total amount of carbon dioxide that we can carry. So, in some animals, we can have this much, and therefore maybe 1/3 of the carbon dioxide carried in the blood is in the carbon dioxide molecules, and then only little bit in other forms. And then in some cases we can have very high concentration of carbon dioxide in the blood, because we can have it in bicarbonate molecules and other forms. And this can be called like carbon dioxide carrying capacity. So the binding and modification is enhancing the carbon dioxide transportation. So if we have high metabolic rate, like we humans, then we're producing a lot of carbon dioxide, and then we can put it in the blood. Yes. And these air-breathing vertebrates, like the human, we have a very high carbon dioxide total concentration. We have to remember that this is not the carbon dioxide itself, it's mainly on the bicarbonate. And then these aquatic animals, the carbon dioxide, the capacity is much lower. So they can carry much less carbon dioxide in their blood than we or even turtles. So that means that, okay, breathing the air as a terrestrial animal is completely different from breathing with the chills. And you can see that over in these very low partial pressures, it's very steep in these fish and other aquatic animals. And the idea is that these aquatic animals, they are operating on a completely different scale of partial pressure. So in this very low partial pressure, it's the only place where they are modified. So that means that if this bullfrog is highly active, it will produce a lot of carbon dioxide. So the carbon dioxide concentration can be 30 millimeters per 100 milliliters. But then this blood is going on the respiration, and then you can easily reduce it. in the blood, because you can get rid of it because it's on this steep curve. Versus in humans, this difference is much smaller. We can carry a lot of carbon dioxide, but it's difficult to get rid of it. But we can a little bit get it easier with the Haldane effect. So depending on how much oxygen we have in the hemoglobin, then this oxygenated blood is carrying less carbon dioxide. So over here we have the oxygen, and over here we don't have oxygen. So that means that, okay, in that case, it can carry carbon dioxide, or then it can be affecting on the binding of protons, etc. And that means that, okay, when we are losing the oxygen from the hemoglobin, the carbon dioxide is forming the bicarbonate, and then this proton can be binded on hemoglobin. And this affects on our circulation quite drastically. So, in the venal blood, the oxygenated, it's only partly oxygenated. So, that's why it can carry much more carbon dioxide. And then when this blood is going on the lungs, this carbon dioxide carrying capacity is reduced. Not only because there is lower partial pressure, but because it can carry less carbon dioxide. So it's easier then to get rid of it. We don't need so steep curve. Okay, so how it works. Okay, here we have red blood cell in the blood plasma, etc. And in the hemoglobin, we are binding oxygen. And when we are losing the oxygen, then this imidate cell group, these certain amino acids, they are binding protons because the hemoglobin is deoxygenated. And how we can get these protons is by the carbon dioxide that is coming from the metabolism. It will go in the red blood cell to either to bind in the hemoglobin itself, but also it can be catalyzed in to form the bicarbonate, and then you have the proton that is binded in the hemoglobin. And how we can form the bicarbonate is because we have carbonic anhydrase in this reaction. And in some cases, we also have the same kind of carbonic anhydrase as a membrane-bounded protein there. So therefore, we can also modify even without the red blood cells to form the carbonic acid and then the bicarbonate. And then this bicarbonate is buffered, is carried inside the red blood cell, and it's also diffused out with a chloride antiporter or ion exchanger, and then it's carried in the plasma. And this bicarbonate is produced in the plasma, and because it's then affecting on the pH, therefore we have other plasma proteins that are buffering these pH changes. Okay, What about then the pH? In humans, we are in quite narrow pH range. So if it drops below 6.8, we are dead. If it rises above 7.7, we are dead. So we try to keep it constant. For example, in humans, 7.4, always. And quite often, in early studies, they were thinking, okay, this is something that happens in all animals. But actually, in the ectotherms, it's variable. So in high temperatures, the pH in the blood is lower. And in the low temperatures, of course, it's higher. And might be that this is caused by the protein binding on the histidine amino acids, but might be also some other effects also. And this pH can be affected on the carbon dioxide transportation, increasing the bicarbonate production. Thank you.